The overall goal of this experiment is to evaluate levels of endoreduplication within potato tubers.
This method can help it answer key questions about potato tuber morphology, such as the developmental stage at which tuber tissues undergo endoreduplication. The main advantage of this technique is that it provides much clearer results compared to typical flow cytometry preparation. Demonstrating the procedure will be Parker Laimbeer, a grad student from my laboratory.
After preparing solutions and buffers according to the text protocol, surface sterilized potato tubers by immersing them in 75% ethanol for at least 5 minutes. Remove the plant materials from the ethanol and allow them to air dry. Prepare and label sterile 50 milliliter conical tubes for each sample and aliquot 15 milliliters of filter-sterilized plasmolysis incubation solution, or PIS, into each. Using either a sterilized scalpel or a cork borer, sample approximately 1 cubic centimeter of the desired sterilized tissue. Use the sterilized scalpel to coarsely chop the tissue into approximately 3 cubic millimeter pieces and transfer the tissue to a conical tube containing PIS. Then, incubate the tissue at 4 degrees Celsius overnight. To generate potato protoplasts, begin by preparing and filtering an appropriate amount of enzyme solution, or ES, with 0.71 molar mannitol, 3 millimolar calcium chloride,